Hello, my name is Vivian Yang. I'm currently a high school junior at Canyon Crest Academy, and today I'll be talking about some of the devices used in that biotechnology. So what is biotechnology? Well, I'm sure you can already tell from the name that it's technology that deals with biology. Well, there are these devices that aid in scientists' research regarding biology, like what we're going to be talking about today, where it will help identify certain things in our DNA. This video will mainly focus on how biotechnology can be used to identify diseases. Now, before we get into the biotechnology aspects of this video, we need to be able to understand the biological processes that lead to genetic diseases and how those diseases are expressed. So let's start by talking about DNA replication. DNA replication starts when the double helix unwinds itself. A primer made of ribonucleotides binds with both strands. It is important to remember that DNA synthesis cannot start on its own. What this means is that the monomers that make a DNA sequence, which nucleotides, cannot just start creating a chain wherever it likes. These monomers need to be attached to other nucleotides to build the DNA, which is why this primer needs to be attached first by the primates onto the template strand before the successive nucleotides can be added to the new DNA strand. These new nucleotides are added one at a time to the new strand by DNA polymerase III, and then the primers are changed to regular DNA nucleotides by DNA polymerase I. The result of this process is two strands of DNA, which will then separate from each other to become two individual strands of DNA. Now, the thing that allows DNA to be replicated perfectly almost every time is due to the properties of the nitrogenous bases of DNA, represented by these squares. Notice how they are two pairs that can only be attached to each other. For example, a nucleotide with a cytosine base can only be paired with a nucleotide with a guanine base. This means that only a guanine-based nucleotide can be added to the sequence when there is a cytosine base on the other strand, since they are complementary. And this feature ensures DNA is replicated identically almost every time. Next, let's discuss transcription, the process where DNA is transcribed into RNA. The DNA unwinds just like in replication, but then RNA polymerase rather than DNA polymerase creates a complementary strand to DNA. The RNA transcribed serves a very important purpose, that is to carry out codes for creating proteins from the DNA in the nucleotides to the ribosomes where a protein will be made. Translation is the next stage, where the RNA is read and a protein is synthesized. mRNA leaves for the ribosome where the protein is made. The RNA sequence is read in groups of 3 known as codons, where the tRNA will bind with a complementary anticodon. And on the other end of this tRNA is an amino acid that is coded for depending on what the sequence of the codon is. These amino acids carried by the tRNA are what forms the protein as the RNA is translated. The amino acids bind together, forming a polypeptide, otherwise known as a protein, that will carry out functions in the body. Translation is initiated when the ribosome reads a specific sequence known as a start codon, which always has the AUG sequence. Translation then continues until it reaches a codon known as a stop codon, which either has the sequence UAA, UAG, or UGA, and there is no amino acid to pair with those codons, which is what stops translation. The amino acid chain then leaves the ribosome, and you end up with a polypeptide that will then be used to carry out whatever function it's meant for. It is important to know that different RNA sequences code for different amino acids that have different chemical properties. These differences between the properties of the amino acids contribute to the shape of the protein based on how those properties interact. Very important to remember here is that the structure equals function. For example, we know how opposites attract. Well, amino acids with positive and negative charges will be attracted to each other, while those with the same charges will repel. Now, there are also other properties like hydrophobic and hydrophilic. with the hydrophobic amino acids typically being on the outside layer of the protein since the hydrophobic amino acids can't interact with water. So why is any of this important? Well, these properties of different amino acids result in unique shapes when the amino acids are sequenced, resulting in a variety of different functions depending on those shapes that they create. A change in protein structure can have significant alterations to what it does. In fact, this concept is the main explanation for why mutations can be harmful to your body, because a mutation causes a change in the structure of the protein and that changes its function. Most of the time, this change will be negative. There are multiple types of mutations in our DNA that can have varying effects on the body. The main ones you should know for the video are missense mutations, where there is one change in amino acid sequence, nonsense mutation, where the amino acid sequence stops earlier than supposed to because of the presence of a stop codon, and frameshift mutations, where the insertion or deletion of a nucleotide shifts the reading frame and all the subsequently produced amino acids. One of the biggest parts of biotechnology is the ability to detect and treat diseases, and that's the main focus of this video. Most of the fatal diseases are found in our DNA, which was passed down from our parents. This is why knowing your family history can help you understand your likelihood of having a genetically inherited disease. If they had it, chances are they probably passed down those genes to you. Now let's take a look at techniques like PCR genotyping, microarray, and DNA sequencing, which are among some of the tools used to aid in the detection of a disease by determining the presence of a specific DNA sequence or the protein the sequence codes for. If you guys remember from earlier, one type of mutation is missense mutation, where one letter in the DNA sequence is incorrect, which results in the wrong amino acid being attached to the protein. Now, we call this DNA sequence SNP, or SNP, which stands for single nucleotide polymorphism. We can use PCR to test if a person has a mutation on a DNA sequence by testing if SNP is present. PCR, or polymerase chain reaction, is a process in which a segment of DNA is replicated multiple times. We do this to have a higher concentration of that segment to work with and identify because a single strand of DNA is tiny. PCR is essentially the method of mass-producing a segment of DNA and is often used for the process by inserting DNA fragments into plasmids. There are 3 stages to this replication process: denaturation, annealing, and elongation. During denaturation, the 2 strands of DNA separate. This is done at a very high temperature, around 94 to 98 degrees Celsius, so just short of the temperature at which water boils, which would make sense because it is at this temperature hydrogen bonds holding the DNA together breaks apart. And why do I mention the boiling point of water? Well, water molecules are held together by hydrogen bonds, and when it boils and becomes a gas, these hydrogen bonds are broken, just like how they break between DNA molecules. During annealing, the temperature is cooled to be around 72 degrees Celsius, and primers are binded to complementary DNA sequences. As mentioned before, a new DNA strand cannot simply be created. It needs to be added onto something or be elongated to something. Therefore, we need to add these primers. The last stage is elongation, where complementary nucleotides are added along the primer. This is the step where the DNA fragment is replicated. We see that each individual strand is serving as a template to which a complementary strand is synthesized. Remember, we talked earlier about how base pairing works and how it synthesizes the same sequence every time because there is only one specific nitrogenous base that can pair with another specific base. For example, cytosine with guanine. A special type of DNA polymerase known as Taq polymerase is used in this elongation stage. Taq polymerase is a DNA polymerase used by microorganisms that live in the extreme environments like hot springs. This DNA polymerase has adapted to be able to function in these extremely hot conditions. One thing to consider about all proteins is that they only function at specific pH and temperature. For example, if the temperature is too high or too low, certain body functions will not be carried out because the proteins in charge of those functions will no longer be able to work because it is denatured. Because of the high temperature required for the previous steps, we need to use a DNA polymerase that won't denature or lose function in those conditions. The DNA polymerase humans use would not work since they function best at our body temperature of 36 degrees Celsius. You can see it's much lower than the conditions we're working with, hence the need for Taq polymerase. So these three stages make up one cycle of polymerase chain reaction. We typically go through about 30 cycles to get around 1 million copy of DNA. Each time we run the cycle, we are doubling the number of fragments we have, so the amount we have is increasing exponentially. So before we run any tests with our fragments, there is an additional step where we need to digest these PCR products. And no, not like digesting food. We're digesting the fragments with restriction enzyme. Restriction enzyme will cut DNA at specific sequences known as the restriction site. These restriction sites are a specific sequence of DNA that the enzyme will bind to upon recognizing it. This step is important because it differentiates SNP from regular non-mutated sequences. The restriction enzyme specifically recognizes the sequence only present in the mutated strand, so it will only be cut in the DNA fragment if the mutated sequence is present. So why do we care that the mutated sequence is cut, but not regular sequences? Well, this brings us to the last stage of this experiment. A gel electrophoresis is used to identify the length of our DNA fragments. This equipment runs an electric current through an agarose gel, in which we'll place our DNA fragments. Now, DNA is negatively charged, and we know that opposite charges attract, so an electric current is created on the side opposite of which we put our DNA fragments in. So we can load our DNA fragments into these wells, the boxes shown in the image. So this side will be negatively charged, so the same as DNA. So when there's a current, the DNA will move towards the other side, which is positively charged. The DNA will start moving towards the oppositely charged side, but not all DNA fragments are going to move at the same speed. You see, the reason we have to put our DNA into this gel is because the gel has tiny holes in it to separate the DNA fragments depending on size. Imagine you're trying to push a ball through the door. If the ball is wider than the door, it'll take you more time and energy to push it through the door compared to a smaller ball. Gel electrophoresis is essentially doing the same thing. It allows smaller fragments to pass through and move more easily and quickly. So the size of the fragment determines how fast and how far it moves from its original location. We are able to use this difference in location to determine which fragment is smaller or larger. So how does this help us identify SNP? Well, remember our previous digestion step and how the fragments are cut if the mutated sequence is recognized. So those DNA fragments that are cut will be shorter than the ones we didn't cut. We will want to test two samples of DNA, one with digested and one with undigested DNA. The two samples should be identical, except one will have been digested with restriction enzyme and the other will not. Supposedly, if the restriction site is present and the DNA gets cut into smaller pieces, our gel electrophoresis will show one lane's DNA to have traveled further than the undigested, uncut DNA. Here we can see that the lane with the digested DNA traveled a lot farther, meaning the restriction enzyme recognized and cut the mutated sequence, and we can identify that the mutation we're looking for is present. Now, if we look at the image, we can see in one lane, the samples traveled a lot further than the other. This is because it contains the mutated DNA, digested and cut by the restriction enzyme. If we see another digested sample, notice how it traveled the same lane as the undigested sample. This is because the restriction site was absent, meaning the mutation sequence was absent. Therefore, the DNA was not cut, so we know that the individual in this sample does not contain a mutation, unlike the individual from the other sample. This technique is also used for DNA fingerprinting. Basically, you would try to match the DNA at a crime scene with the DNA of the suspect. And you try to identify which of the suspect's DNA most resembles the one found in the crime scene. This helps forensic scientists identify who was responsible for the crime, just like how they help scientists determine if a mutated gene is present. Probes can also be used to find specific pieces of DNA. Remember that for a trait to be expressed, transcription has to be initiated to create mRNA, which will later be translated to make the needed protein for expression. Also remember the concept of transcription creating a complementary strand to the template DNA, meaning the RNA sequence would be able to bind with the DNA sequence. Because of this, we can use mRNA to locate the DNA since the two strands can bind together. This is where probes come in. Probes are oligonucleotides, a short sequence of DNA or RNA, and these sequences of DNA can help identify genes of interest by binding or hybridizing with sequences of genes of interest. By marking the probes with a fluorescent or radioactive label, we will be able to identify the location of the gene once it binds with the DNA at the location of the gene of interest. The benefits of using probes to identify sequences is that it can not only help us identify the presence of a mutated sequence, but it can also help us identify any gene scientists are looking for, which they can then extract and have it be used for recombinant DNA or gene therapy. Microarrays is a device that utilizes probes for gene identification. How it works is that first, RNA from a healthy and mutated cell are extracted, and using reverse transcriptase, a process where RNA is transcribed into DNA, cDNA is synthesized and fluorescently labeled. The cDNA that came from each cell are labeled with fluorescence differently, and you'll see why soon. These cDNAs are then put into wells with probes that are a specific gene. The cDNA will either bind or not bind with the probe, and whatever does not bind will be washed away. This leaves our well with only the cDNA that had binded with the probe, and we can determine whether the cDNA from the healthy or mutated cell had bind with the probe by measuring the degree of fluorescence that corresponds with a specific cell type. This allows scientists to determine the gene expressed by the cell, depending on whether the cDNA binds to the probe or not, and this helps scientists identify whether the cell they obtained the sample from is cancerous, depending on what genes were expressed. And by understanding what those cells are expressing that is harming the body, and the identification of those proteins produced, gives scientists a target protein to destroy when they are trying to subdue cancer's effects. DNA sequencing techniques allow scientists to be able to identify mutations and compare their DNA to wild types. This is another way for scientists to test for the presence of a disease. This genetic test allows scientists to more closely study and compare the sample gene, even to identify the type of mutation it's causing. There are three methods for sequencing: Sanger, high throughput, and NextGen. The two methods I'll be talking about are the Sanger method and NextGen sequencing. Sanger sequencing relies on the use of ddioxynucleotides, also known as ddNTPs. The difference between ddNTPs and regular nucleotides is that they are missing an oxygen molecule on their carbon number 3. Typically, a hydroxyl group is located there, as seen in the image. This allows the nucleotides to bind with each other to create the chain that is the backbone of the DNA. The change from OH to H prevents the binding of an additional nucleotide, so the strand can no longer grow. So in ddioxynucleotide sequencing, a few of these ddNTP nucleotides are put in the solution along with regular nucleotides while synthesizing a complementary DNA strand. 4 tubes are set up so each has all the necessary reagents for DNA synthesis, like the template's DNA, primer, deoxyribonucleotides, but also ddNTPs. Each tube also only has one of the 4 nitrogenous bases. This process relies on the random chance of one of the ddNTP attaching to the growing strand rather than the regular deoxyribonucleotide. And the location for where the chain stops growing is used to determine the base at that location. Remember, each tube's ddNTP has one of the 4 possible bases. So let's say the ddNTP in one tube only has cytosine base. Well, that means the location where synthesis stops, there is a cytosine ddNTP. And that's how scientists are able to determine the location of each base. All the newly synthesized strands are put through a gel electrophoresis to compare the length of each strand, depending on which base they stopped at. This is how scientists are able to piece together the sequence, by comparing the length at which each sequence stops and what base it stops at. As you can probably tell, this process is very complex and very time-consuming, requiring multiple copies of DNA just to determine the location of one of the millions of bases on DNA. So now we move on to next-gen sequencing, also called massively parallel sequencing. And this method is much more accurate compared to standard sequencing and only requires one DNA sample for analysis. There are multiple next-gen sequencing systems, but I'll just be discussing the one used by the Ion Torrent Personal Genome Machine. How it works is that the DNA sample is fragmented and amplified through PCR and then attached to millions of beads that are then separated into wells. These wells are flooded with a solution with one of the 4 types of nitrogenous bases, which binds to the fragment on the bead. When free nucleotides bind with a nucleotide on the template strand, a hydrogen ion is released, and this causes a change in the pH of the solution in the well. The change in pH is converted into voltage, where it is measured to indicate the binding of that specific nitrogenous base. This process is repeated with a different base solution, and scientists are able to sequence the DNA by recording what base was present in the solution that caused a change in the pH as a result of the binding. And the process is repeated multiple times until the scientists achieve the desired length or all the sequences of the DNA that they're looking for. So that about sums up the video. So to recap, we talked about 3 different ways we can use technology to identify diseases. PCR genotyping works by creating multiple small fragments of DNA that are digested with a restriction enzyme. Samples are then put through a gel electrophoresis. Based on the size of the fragments, we can identify the presence of a gene, since it would have been cut by the restriction enzyme. Microarrays work by measuring the degree of fluorescence to determine if a cell has a specific gene. And lastly, DNA sequencing works by sequencing the nucleotide bases of DNA to allow scientists to closely study the variations of DNA sequences and identify harmful ones. As our understanding on how gene expression and genetic processes in our body work, we can use that to aid the progression of biotechnology to help cure diseases, starting with our ability to identify them. Without scientists' understanding of how genetic information is conserved and passed down, how mutations emerge, and cellular processes that edit RNA, the technologies mentioned today would not exist. It is by understanding how biological processes occur in our body that scientists are able to use this information to help find treatments to inhibit diseases and enhance our immune response. Thank you for listening to my presentation. Hope you guys learned something new about biotechnology.